Amelogenesis is the process of enamel formation which starts with the deposition of enamel matrix by ameloblasts followed by the mineralization of the matrix. Hello, salam, I am Dr. Usman Ali Khan. Enamel matrix essentially consists of organic material composed of two groups of proteins, namely amelogenins and non-amelogenins. Non-amelogenins consist of proteins enamelin, tuftelin and ameloblastin. The amelogenins form the bulk of the enamel proteins comprising at least 90%. And once enamel is fully formed, organic material makes up only 4% of the enamel and the inorganic portion of enamel is composed of hydroxyapatite and comprises of 96% of enamel. Amelogenesis is divided into three stages referred to as the pre-secretory, the secretory and the maturation stages. The pre-secretory stage is the phase in which inner enamel epithelial cells differentiate to become ameloblasts that are ready for secretion. In the secretory stage, ameloblasts secrete organic matrix comprising various enamel proteins forming the entire enamel thickness. Ameloblasts carry out activities in the maturation stage that help in degradation of enamel matrix and subsequent replacement by minerals or inorganic material. Thus completing the process of enamel formation. The pre-secretory stage is further subdivided into two phases referred to as the morphogenic phase and the differentiation phase. In the morphogenic phase, the ameloblasts are formed. In the morphogenic phase, the enamel organ is in the bell stage of root development and the shape of the crown is established. The inner enamel epithelial cells are low columnar or cuboidal cells with a central nucleus. Golgi apparatus is located in the proximal portion of the cell, that is towards the stratum intermedium, and the other cytoplasmic components are scattered across the cell. During the phase of differentiation, the inner enamel epithelium starts to differentiate to become an ameloblast. The cell elongates and becomes tall columnar and the nucleus that was previously the center shifts proximally. Golgi complex shifts distally, that is towards the dental papilla, and mitochondria that were scattered begin to cluster proximally. Now when these changes to the inner enamel epithelial cells are happening, there is simultaneous differentiation of the cells in the dental papilla to Odin blast. Thanks to the signaling molecules from the differentiating inner enamel epithelium. Odontoblasts begin to form dentin at the end of the differentiation phase. In summary, the inner enamel epithelial cell at the end of the pre-secretory stage differentiates to become an ameloblast. with the nucleus polarized away from the dental papilla and most organelles located distally. Ameloblasts are attached to each other with the help of junctional complexes, both proximally as well as distally. Once the predentin or the first layer is formed, the ameloblasts are separated from the dental papilla. The second layer of dentin is formed. It stimulates the ameloblasts to begin enamel formation. Now it has to be understood that this initial layer of dentin is necessary for stimulation of enamel formation. Just as it was necessary for the inner enamel epithelium to help in differentiation of dental papilla cells to odontoblasts. In the secretory stage, ameloblasts develop a structure called the Tums process. Now a fully developed Tums process is a distal extension or projection of the ameloblast consisting of a proximal portion and a distal portion. However, at the beginning of the secretory stage, Tums process has only a proximal portion. Ribosomes help in synthesizing proteins which are then packaged into secretory granules in the Golgi complex. These secretory granules containing enamel proteins are then secreted via the Tums process. The initial layer of enamel matrix is laid down on the mantle dentin and is mineralized immediately. Hence this initial layer of enamel does not contain enamel rods. Now as more enamel matrix is laid down, the ameloblast moves away from the enamel with the Tums process developing an extension from the proximal portion. This extension is the distal portion of the Tums process and it keeps elongating as more enamel matrix is secreted. This is the initial layer of enamel matrix secreted with the distal portion penetrating the enamel. Now it has to be understood that rod and interrod enamel is a result of the Tums process. So let's understand how this happens. When the distal portion of the Tums process is developed, secretion of enamel happens from two sides. One is from the proximal portion of the Tums process and the other from the distal portion. The enamel matrix secreted by the proximal portion encircles or surrounds that enamel matrix secreted by the distal portion of the Tums process. The enamel matrix secreted by the distal portion of the Tums process forms the enamel rod, and that which surrounds the rod is called the As the enamel matrix continues to be secreted, not only does the distal portion of the Tums process elongate, but also gradually becomes thinner and gets squeezed out of existence. Once this happens, there is a thin space at this junction between the rod and interrod enamel that is filled with organic material to form the enamel sheath. Once the distal portion of the tome's process disappears, the ameloblast again resembles how it was initially, and thus the last increment of enamel formed has no enamel rod. Hence it has to be understood that the final enamel is composed of two parts. The first part is composed of rod interrod containing enamel squeezed between two layers of rodless enamel. The maturation phase of amelogenesis consists of a transitional phase and a maturation proper phase. Before the enamel matrix could mineralize and mature, there is a brief transitional phase where ameloblasts reduce in height and volume. Also, ameloblasts undergo programmed cell death or apoptosis. And there is approximately 25% reduction in the number of ameloblasts in this phase. The maturation phase is when the bulk of the proteins and water in the organic matrix are removed to be replaced by inorganic material. This is accomplished by a process called modulation. Modulation is a process of cyclic creation, loss and recreation of an invaginated, ruffle-ended apical surface. That is, ameloblasts cyclically alternate between having a ruffle-ended border and a smooth-ended border at the apical surface facing the enamel matrix. A ruffle-ended ameloblast possesses a tight distal junction and a smooth-ended ameloblast possesses a leaky distal junction. Ruffle-ended ameloblasts possess enzymes like lysosomes, metalloproteinases and serine proteinases. These enzymes are secreted via their ruffle-ended borders to degrade in bulk the proteins in the enamel matrix. Once the proteins degrade to small fragments of peptides, they escape and diffuse into ameloblasts via the leaky junctions of the smooth-ended ameloblast. When the enamel matrix is being degraded, the calcium-binding proteins and calcium adipose is present in the ameloblasts. Help in pumping calcium ions to the degrading enamel matrix in order to mineralize it. And help in active crystal growth, thereby fully forming enamel.